Hey everyone, welcome to another edition of Metaflow Office Hours. We have an exciting session coming up today. Today we have Siegfried Gessulat as our distinguished speaker. Siegfried is the head of machine learning at MSAID. MSAID is a bioinformatics company that was spun off from the chair of proteomics and bioanalytics at the Technical University of Munich in Germany. The company was founded by an interdisciplinary team of scientists with the vision to provide better computational solutions to the field of proteomics. And if some of you are like me, who are curious about what is proteomics, I'm sure this talk will give us some idea about that. Siegfried has been with the company for almost 5 years now. He has a PhD in biochemistry, biophysics, molecular biology, and computer science from the Technical University of Munich in Germany. Siegfried and everyone else, welcome to Metaflow Office Hours. Over to you.
Thank you so much for that great introduction, Sri. It's my honor to be here. Thank you. I'm very excited to talk a little bit what we do at MSAID and how we use Metaflow and the Out-of-Bounds platform. Let me share my screen real quick. Okay, I hope that works. Yeah, okay. First of all, I would like to give a shout out and thank you to Hugo from Out-of-Bounds, who supported me in with this presentation, because we showed a shorter version of this at a meetup here in Berlin together. So he provided all the nice looking graphics and drawings here. So it's really nice. Thank you to Hugo. Sri already introduced MSAID. Thank you for that. I'll just give a little bit more context at this beautiful picture here of the 20 people, roughly 20 people that we are. We have offices in Munich and Berlin, and we are concerned with mass spectrometry-based proteomics data, and I will talk a little bit what that actually is and why we are so excited about that. Most of our team regularly works with that data. So 10 of the 20 people on a daily basis do data engineering, data science, and machine learning. So we regularly look at the data, question the data, and transform the data, and then train machine learning models with them. So that's very important to us. Let me give a little bit of an introduction what mass spectrometry-based proteomics as a field is. And then I'd also like to show you a little bit how the data is actually looking like. Proteomics is the science of studying proteins. And we are interested in proteins because they are little molecular machines. You can think of that as molecular machines that carry out all work to sustain life. On the right-hand side picture, you can see a beautiful drawing of a cell, and lots of biology is happening currently at that cell, and most of that is done, that work, that what's actually happening is done by proteins. For example, they catalyze energy production, carry oxygen from lungs to the rest of the body, and also signal information and messages between cells. So they provide structure, function, and regulation in cells. Mass spectrometry is the technique that lets us measure proteins from biological sample, and it's a standard high throughput technique. And the combination mass spectrometry-based proteomics is, for example, used in drug discovery research for complex diseases such as cancer. So how does the process and the data actually look like? So in principle, you start with a biological sample, cells, you extract the proteins out of the cells, then you do something called a digest. The protein is basically a long string of characters, amino acids, and you cut that into pieces, and the smaller pieces we call peptides. Those peptides are then, by technologies called liquid chromatography and electrospray ionization, are charged and fractionated across time, and this is how they enter the mass spectrometer. You can see the first plot on the left side here. There is different amount of signals coming into the mass spectrometer at different point in time. At the beginning, there's nothing. Then you see a little more signal coming in, and the signal is dropping off again. The first measurement that we are interested in is called MS1 scan or measurement. In that, we isolate a certain mass range where we measure basically the mass of a peptide. So we have the mass of all the amino acids of the peptide, and we measure that as a whole and isolate that. Then, in a second step, what we do is we take that isolated peptide and fractionate it. We basically shatter it into smaller pieces, into substrings, so to speak. And usually these are prefixes and suffixes of the small sequences, and those are called, we label as B and Y ions in the plot on the right side. And in the second scan, in the second measurement, the MS2 scan, we measure the masses of these smaller pieces to, from there, infer back what peptide sequence could have given rise to the MS2 and MS1 scan. So the computational workflow is actually going backwards from the MS2 scan. We try to match different peptide sequences to that measurement, then score those different peptide sequence, peptide spectrum matches, and then do error control and identify the best scoring match, peptide sequence match to that spectrum given some error threshold. And then with that, we can do quantification. So this concludes my little overview of how the data looks like. But I'd briefly like to talk about machine learning, why we're needing machine learning. So actually, we can apply machine learning at every step of this computational workflow. So matching, scoring, error control, quantification. And here are some picked use cases that you're probably familiar with. So for example, we can predict properties of biomolecules, for example, regression or density estimation. And we can use these predictions as a prior in our data analysis to make the data analysis better. Another very important thing is to calibrate our workflow to specific laboratory environments. The laboratory environments are very different from one another, and we use fine-tuning and regression to basically calibrate the workflow or the data to improve the analysis. And then in the error classification and control step, in the field, usually we do semi-supervised classification to improve error classification and reduce false positives. Unlike many other fields, we are very lucky that public proteomics data is abundant, but we still have a problem because the harmonization is very challenging. I mentioned already that across different laboratories, there are very different environments, and those have many reasons. For example, the measurement methods are not standardized. They can be vastly different from lab to lab. There are many different analysis software and output formats and assumptions made by the software, which makes the output data very different. And then reference standards are also not used consistently across the field. So one publication might use this reference standard and the next one uses a completely different one. So a common reference standard is not present in the field, unfortunately. So our main work when we want to work with the public data is actually the data harmonization.
So question,
yeah, sure.
Question here. Is it okay to ask questions right now? Or
yeah, please go ahead.
so in this case, so especially when you talk about the data harmonization and how the methods, the measuring methods are different, the analysis software is different. You're trying to harmonize it among all the experiments that you can control. This is, and I'm guessing if with that you can probably set a standard that others can also follow. Is that correct?
That is correct. So so I can maybe give the next slides are
Okay.
two actual use cases,
Mm-hmm.
machine learning use cases, and then I can answer that question based on that specific use case. If
Okay.
that's okay.
Sure.
Yeah.
Sure.
Cool.
Yeah. Of course.
So the first use case, and I mentioned that briefly already, is predicting the fragmentation pattern. At the top on the left side, you can see again a measured an MS2 spectrum. So we have the measured intensity and the fragments on the x-axis. The MZ is basically the mass of the fragments. And usually what we do to identify which sequence this is we just try one peptide sequence, the string above the measured spectrum, and we calculate all the possible prefix and suffix masses of that string. And then we can match this theoretical red spectrum at the bottom to the actual measurement at the top. But this is just a calculation. There's no machine learning. We know the atomic masses, so there's no machine learning. But what we would do here is to actually, what we do in our software is we predict this probability density, basically. And then we have two axes where we can score the quality of the match. And this would basically be a perfect match. To come back to Shree's question, so how would we actually calibrate data that looks different from different experiments or from different publications? We identified parameters that influence the variance of these measurements, basically. And what we do is we align measurement data to our machine learning models. So we have basically a reference machine learning model. That machine learning model can predict all potential peptide sequences, how the spectrum should look like. And then we can ask the model back, okay, what input parameters except the peptide sequence look most like the measurements that we have in hand. And then this is basically how we harmonize based on this other factors, external data. So we line to machine learning models. In this case. There's a second case, which is retention time prediction. This is much simpler. It's a retention time, it's a regression task. So we want to predict when in an experiment, which could range from 7 to 180 minutes, we expect a peptide to appear in the measurements. So peptide sequence in and some time estimate out. How do we do the data harmonization in this case? Well, first of all, what we don't like is that the experiment, the maximum, the time ranges are vastly different, right? 7 minutes or 3 hours. So what we do here is we normalize this to a z-scored retention time, so a time-independent measure, basically. And then we do, for each file that we analyze, we do a transfer learning. We have, from a first look at the data, we know the minimum and maximum time, and then we can scale back to the actual experiment.
Cool.
So, right. Yeah, so these are just two picked use cases. Yeah. And sorry, is there another question?
No.
Okay, cool. And then I would basically give an overview where we would, why we're using Metaflow and where in-house in our data preprocessing we use that, or processing in general. And I have 3 slides, and one is basically where we came from. which we're seeing here. So that's the past. I have a status quo, and then I have a third slide where we actually want to be, and that will be in the future somewhere. So in the past, we had a lot of the stuff, the data harmonization, training, and model evaluation was all custom pipelines because our data is so custom, right? There are no bigger frameworks that do all the stuff that we want to do. So it was a lot of like custom scripts, and they were glued together, and we had a database that kept track of all the data transformation runs, and we shared the data across different workstations via shared drives. That worked okay, but maintenance, reproducibility, observability, scalability were all problems because we needed to maintain all of that ourselves and needed to make sure when we upgrade databases, when we updated database schemas or our software, that everything was working together. So I think we worked together with Outer Bounds now for around a year and used the Outer Bounds platform in conjunction with Metaflow. And we migrated basically all of our data harmonization and evaluation scripts to Metaflow and run them on Outer Bounds platform now. Which is the most critical part of our code base in the machine learning part. And I would assume that is around 80% of the lines of code that are now run by Metaflow. The benefits that we see so far are the scalable infrastructure. Currently, we could only scale to the limits of one workstation, and now we can scale what AWS allows us, basically. We are also super happy with the observability. The API and the UI, we use both regularly to make informed decisions on new trainings and data quality. It's much easier also to restart and debug. And another thing is that people really like to work with Metaflow, so the onboarding also was much faster now. Where we want to go is to eliminate some of the other pipelines that currently use different technology, because for the data acquisition, although it's not super complicated, it's also not currently super critical, but there is no reason why we would use Argo over Metaflow in our specific case, and would just make things easier for us if we have a streamlined technology stack. Same for model deployment. Currently, there are several manual steps involved to actually bring a model into production, to tag or push models to the right places. Obviously, that's also something that we would like to automate very soon. And we are excited about the events and gluing together different pipelines or flows together via events for that. Right. Yeah, I think that was the tech stack. And then I have one overview slide basically on how what the main benefits for us currently are. So I already mentioned that the scalability is a huge plus for us. And currently we are able to ingest proteomics data faster than it's published to the public repositories. That's great. So we can basically ingest everything that is newly published. And we were able also to retrospectively ingest all public data for the specific set of peptides that we were interested in this year, and could make use of that for training our machine learning models that we will release within the next month. These data sets are basically are based on 5 terabytes of data from 50 different repositories. Or actually, it should be more by now. This is an old figure. But so we are super excited about these numbers. And we could speed up our processing speed. So usually this would have taken us, so 5 terabytes would have taken us maybe a week to process, 1 to 2 weeks, and now it takes us half a day or maybe a day. That depends a little bit on the data. And we, for our people, on the people side, we could reduce the onboarding time substantially. So one colleague, she was able basically within her first week, starting with us, to write code that actually transformed data and created data sets for us. So that's really nice. And people are very happy with the structure that they get from Metaflow. If I would have to guesstimate how much productivity we gain from the people, I would say that one FTE is basically free from managing the platform and the infrastructure that we needed to run that, and can now focus actually on real data science and machine learning to actually generate value for us. So that's that. Yeah, I think that's what I wanted to show, and I'm happy to take more questions if there are any.
Yeah, this is awesome. Thank you so much for sharing this. Can you share a little bit about kind of you mentioned 5 repositories or 5 terabytes of data and 50 repositories. What is this? What is a repository? How do you like how do you get it? And it's a public data. So is it in a, what does that public data mean? Like, is it a public S3 bucket somewhere from where you download it, or what is it?
Yes, actually, that's a very good description of it. Yeah, so basically, our field decided that, or agreed that there are basically 2 central places where we store all data from all publications. So you cannot publish results in our field without storing the corresponding data publicly in this repository. And yeah, this is basically, like you guessed, it is underlying is basically big S3 bucket. Everything is.
I see.
With a little bit of
what
infrastructure around it. Yeah.
Sure. And what is the data? Like, is it, I mean, so you talked about kind of sort of protein sequences being strings, and is it like a giant file with lots of like binary files with lots of binary strings in it? I'm sorry, these are two naive and mundane questions,
that's
but
great questions. Yeah. So the data, the vast majority of the data is actually the more or less raw signal that comes from the mass spectrometer. So it's spectra. So it's these MS1 and MS2 scans that you saw. I can maybe just share again.
First or second slide. Yeah.
Right, let me share that again real quick. It's better to have it here. So so actually the MS1 and MS2 measurement here, the 2 right scans, this is the bulk of the data, and this is basically binary data. You can think of this as like float arrays.
Yeah.
And that's the vast amount of the terabytes. And the identification we do in our pipelines ourselves. So we look at that data and then identify the peptide sequences out of that. So then we have for each spectrum a sequence, a string that is matching, and then we do data transformations to actually generate training data for the models.
Got it. Okay. So there is a public, essentially an S3 bucket somewhere. You can download this data from there, which is basically binary data, and you can kind of sort of deconstruct it into strings, and then run a pipeline that will create the raw data for your machine learning model, and that will then trigger another pipeline which actually creates the model, that's where you're
exactly
like, this
so
raw data requires input to that.
Yeah, exactly. So we have several. Maybe I can also show you that. So I have one backup slide that describes this process actually. Okay. Can you see my screen again? Is that working?
Yes. Yeah, this is awesome.
So at the beginning, there are these two repositories called Massive and Pride,
Got it.
which are S3 buckets essentially.
Yeah.
And
Yeah.
then we download that to our internal S3 bucket,
Yeah.
do a search in Argo currently. Then we get the identifications, so spectrum2 sequence matches. Then we do several pipelines, annotation. This is basically determining which of the
Yeah.
ions are prefixes and suffixes. We do the data calibration, what I mentioned with aligning to a machine learning model, and then filtering for data quality. This is also like very specific and a super important step. And then we generate training data, do the model training, and then we do model evaluation.
Awesome. And how frequently is this kind of sort of the raw data in Massive and Pride? Like how frequently does that change or get updated? Or is it like a continuous thing, or is it like, it's an event that, oh, new data has been published once a quarter or something like that?
Yeah, it's basically whenever somebody in our field publishes in some scientific journal. So
I
it
see. I see.
happens a couple of times a week, essentially.
Okay.
So
Okay.
yeah. Yeah.
Interesting. So it is fairly frequent, where if it's a
Yeah.
couple of times a week, I mean, depends, but like, yeah, it's not like it's a one-off thing that happened and then months later some new model got created. You could be creating models, or you could be cleaning up data, processing the data, creating models few times a week, couple of times a week, few times a month.
Yeah, we could do that. I mean, it also depends on how large scale the studies are. So
That's
that
right.
there's also a big variety. So sometimes there are smaller, very focused studies where we have like 10, 20 files that look at a very specific problem, which might be interesting to include in our machine learning model, but wouldn't trigger us to
Sure.
redo the training. Whereas there are other very large scale studies with thousands of files, terabytes of data. They don't get published very
Sure.
often, so maybe
Sure.
once a year or two
Sure.
times a year. Yeah.
I see. Interesting. And then when you create these models, like do they, like how, I mean, is it like a massively parallel job? Or is it more like, give it time? Like I think you mentioned 7 hours in some cases. So limited resources for a long period of time, or is it like very parallel? So you'll fan out to like, thousands of parallel nodes and or parallel tasks, and each of them is doing maybe finite amount of work, but it can go on but it's massively parallel. That's what takes 7 hours. How do you distinguish between
Yeah,
those two?
so yes, it is actually massively parallel. So in recent runs that we did, we basically had like a couple of thousand files in parallel, and each of the steps of this pipelines can be parallelized to the file level. So one file would be 1 to 10 gigabytes of raw input data, and then we would go through the steps of this workflow in parallel for thousands of different files. They don't like the way the cluster is set up currently. We don't like scale to thousands of
Sure,
nodes,
sure, sure.
but Metaflow helps us with that. So
Yeah,
it's very
yeah,
nice that we can just say, okay, here are
yeah,
all the tasks that we want to get done, and then Metaflow schedules that for us, and then
yeah,
we wait some time.
yeah, yeah. When I said thousands of, I meant like tasks. So the task
Yeah,
parallelism can be quite a bit. Underlying the nodes can be depending on the availability, depending on whatever the size of the cluster and all that. It can be limited in size. So
yeah.
that's excellent.